Today's class is on homology modeling. So it's a new term that you are hearing. So what it means, it's composed of two words. One is homology and another is modeling. Now what you do here is that the protein sequence is going to be modeled into the structure. Okay, so on what basis? On the basis of homology. So this is comprising of modeling the sequence into the structure. So that is a model that you're creating based on what? Homology. Homology of sequences to the structure database. So homology is been there since a long time, and what is this graph showing you is how much of an identity is required to make this model. So is it say 30% or is it 80% or 90%? Here it says that almost more than 30% identity is ideal for making a good homology model, because anything less than 30, you see, you won't find any model. So anything more than 30, say 50, 60, more the better, but nothing less than 30. So 30% of the sequences should be identical to the structure. In that case, it is possible to get a good homology model. Now, why do you need to know this information about the protein structures? Now, protein performs a variety of biological tasks, and we know that it can even determine the function of the particular structure. It is more conserved than the protein sequence and more closely related to the function. Or in other words, the sequence to structure relationship gives an insight into the function. So how can we know the function through the sequence? Is by building a homology model, or in other words, the structure, the three-dimensional structure of the protein. Modeling the protein structure has been there since many years, and when you talk about protein folding, so it has been always discussed with reference to the protein folding problem, because protein folds into a unique three-dimensional structure under certain physiological conditions. It could be in the the pH that is surrounded with. So these are the factors. Other factors could be the forces that are present in that environment. So because of certain parameters, it folds in a certain unique way. And this protein folding is what is unique about this modeling process. Now, prediction of the structure is based on a highly similar structure, and when we have the unknown structure, what is required to be done is align the model and the template sequence. So model is the one that we want to determine, and what we need is a template. Template is a thoroughly investigated structure which is available in the database. What we will get by doing this alignment is a completely solved protein structure. What is the next step is, once the alignment has been created, we are going to copy the backbone and the conserved residues for finer alignment and finer development of the three-dimensional structure. So first what we get is a rough structure, and then we go and finalize the backbone and the conserved residues to get the perfect structure. So this adding side chains and molecular dynamic simulation are all finer points of developing the structure. So in this way we can get a fairly accurate homology model. So to explain to you in easy ways, the entire homology modeling can be divided roughly into 8 steps. First is the recognition, the template recognition. And what do you mean by template? Is the completely solved structure which is there in the database. When you align it, that forms the basis, fundamental of developing a protein model. What constitutes this template recognition and initial alignment? So as we saw in the previous class, we can blast the sequence against the PDB. So this helps in actual recognition of the initial alignment to the solved structures present in the PDB. So this is one important method that we studied in the last class to recognize the correct template, because your accuracy of the model will highly depend on how accurate your alignment is, your initial alignment is. For that we need a perfect structure, and BLAST is one method where we can find your recognizing your template. So when you BLAST it, you will get probably the best hit, and you can choose the best hit. Once that is done, there are various servers which will help you with actually developing this protein model. So you have to copy paste the sequence into certain servers which are specialized for this function based on your suggestion of using this particular PDB ID. Next step for this is the alignment correction. So wherever there are gaps in the alignment, we can make a perfect structure, and this will go a long way in developing an accurate model. So the alignment correction is based on finding functional residues which are conserved. And what is the meaning of conserved? Is that they are present in almost all the residues, hence they are conserved. What we can do here is use multiple sequence alignments and do the alignment correction. So we can compare it with multiple sequences and align it and finally develop the correction. If there are deletions, we can shift and make gaps, accommodate for gaps, and this is the basis for alignment correction. So here you can see a gap has been added because it is not exactly aligning with the sequence. Such type of alignments are possible, and the shifting of the gaps is an important step in alignment correction. So here is an example of multiple sequence alignment where various sequences have been compared and the optimum alignment positions have been placed. And you can manually choose which is the best alignment. Now, which are the core residues become the conserved ones, and as shown you in the previous slide, the multiple sequence alignment have been used and the known structure has been matched. So when this has been done, we get a backbone of this sort. The backbone of a small segment is shown in the given example, and this is the actual alignment. So you can see here the given, which is given in dots, they are actually the gaps. Gaps have been matched and it has given to form a large hole. So in actually it is not matching exactly where you can see the dotted lines. So this will result in large holes in the protein structure. Hence we need to take care of this aspect. In the third structure, which is given in blue, you find the gaps are the least. So this is what you're actually doing in the alignment correction method. You are aligning different models and choosing the one where the gaps are less, because gaps are equivalent to large holes in the protein structure. So I guess this is explained properly for you, which is the correct alignment. The structure given in blue is the correct alignment. Now third step is actually forming the backbone generation. What do you mean by backbone? You have studied in your biochemistry, a structure has a certain backbone, the carbon to carbon backbone, and making the model also involves this backbone generation. So the backbone has been copied from the template to your model, and you can make deletions as discussed in the previous slides. Keep only the conserved residues, and lo and behold, your backbone is done. The next few steps I'll explain very briefly, because in another class we can go into the details of it. What is this loop modeling? Loop modeling is taking care of those sequences which have not been assigned a structure. So once the backbone is done, then you would fill in the remaining regions with loops, and that is known as loop modeling. So what happens in the loop modeling is it will consult a library, because those regions have not been formed properly. It will consult a loop library. Library is something like, just as if you don't have knowledge, what you do is borrow books from the library. Here also, the coordinates of the regions which are unknown are borrowed from the library, which is known as a loop library, and the correct assignment of sequence to structure has been done. After the loop modeling is what is known as side chain modeling. So once the loops have been taken care of, we need to perform what is known as a side chain modeling. So here the accuracy of the side chain is important in giving you the correct steps. So the side chain modeling is, you know what is a side chain, and the exact angle in which they occur has to be determined by this side chain model. So in this particular diagram you can see it has borrowed the same rotamers from various libraries, and the one which exactly matches is going to be your correct side chain. So when you're borrowing books, same thing you do is try to pick from various books which suits you, and here also it is going to pick the rotamer which is highly accurate based on the backbone conformation. So once that is done, the side chain modeling is done. What is the next step? Is the optimization step or model optimization. So here all the energy minima has been calculated.